Good afternoon, everybody. Welcome back to the world of viruses. Or maybe you were still in it, right? Who knows? Now, the rest of this course is about how viruses cause disease. And today we're going to talk about immune defenses. This is an outline of the defenses that we're going to talk about. Defenses are sequential, they amplify, and they're specific. And here I divide them into three general areas. The physical and chemical defenses, which we actually talked about last time. The skin, surface coating of tissues like mucus secretions, low pH, etc. These are physical and chemical defenses. They're not specific, but they're an important front line. Then we have frontline defenses, which we'll talk about today. The intrinsic defense systems. And in fact, Dr. Silverstein next week will get into these a little more, induction of apoptosis and other of these intrinsic defense mechanisms. And then finally, the innate and adaptive defenses, the defenses that are specific for microbes, the elaboration of cytokines, inflammation, and the production of defenses that are specific for the pathogen, antibodies and cytotoxic T cells. So here's another way to look at what happens as an overview. You have a pathogen that invades a host. If it's not recognized, nothing happens. But initially, there are what we call pattern recognition receptors. So assuming the pathogen gets past low pH or skin or whatever, it can be recognized, and we'll talk about how that works. And that recognition leads to the innate defense system, which comprises cytokines, dendritic cells, NK cells, and the complement cascade. These may succeed at getting rid of the infection. If not, then we move on to the adaptive response. And the adaptive response produces defenses that are specific for the pathogen, antibodies and T cells, and of course also memory. Assuming you survive the infection, there's memory produced so that the next time you encounter this pathogen, you respond quickly and specifically. And if you remember anything from today, please remember that these defenses don't work by themselves. They communicate. So if the innate defense system encounters a pathogen and can't handle it, it passes it on to the adaptive system. And it communicates with chemicals, cytokines, and with cells. This is another way to look at these defense systems. There are three walls. If you get over the first, which is your intrinsic defenses, your skin, low pH, apoptosis, and other components that Dr. Silverstein will tell you about, then the innate system is signaled. If the virus gets past the innate system, then it encounters acquired immunity. If it gets past that, out of luck. Depending on the pathogen, you either have a bad infection or you're dead. So three stages of infection. So let's first look at the innate immune response. The things that happen before this, intrinsic defenses, Dr. Silverstein will talk about. It's a little out of sequence, but it goes better with what he's going to talk about, the evasion of those specific defenses. So we're going to skip that for now. The innate immune response is something that's always present. You don't have to have an exposure to a pathogen for it to be there, and it's activated very quickly after infection, minutes to hours. It's the only defense available right away. In the first few days, in fact, there aren't specific defenses except the innate defense system. And the innate system comprises cytokines, sentinel cells, dendritic cells, and macrophages, complement and NK cells, which we'll talk about. And it is activated when it recognizes patterns on the microbe, a pattern that is specific for the microbe and is not self. And these are motifs that are only found in microbes. And as I said, if the innate system can't handle the infection, which it does very well, most infections are handled completely by the innate system, but if it can't, it hands it off to the adaptive system at a certain threshold. So let's talk a little bit about how recognition occurs. How does it recognize a pathogen and not yourself? And this began back in 1980 when two scientists in Germany were studying Drosophila. They were trying to find genes that were important for the development of the Drosophila. And they had a particular fly that they had made mutations in, and it had a very interesting phenotype. And the story goes that they looked at it under the microscope, and one of them said toll, which is like cool or far out in German. And so they called it the toll gene, and they got the Nobel Prize for that in 1995. In 1996, it was found that this toll gene actually had a role in immunity in the fly. Flies only have innate immunity. They don't make antibodies or cytotoxic T cells or B cells. And this toll gene not only had an important role in development of the fly, but also in defense against fungal infections. And then in 1997, the next year, similar genes were found in mammals. And this really begins the whole understanding of the innate system. Before this, it had been postulated to exist, that is, a very early defense system, but no one understood how it would recognize the pathogen and not the host. And it turns out that these toll-like receptors are the key. So toll-like receptors are type 1 transmembrane protein. They recognize patterns specific to pathogens. Sometimes we call these pathogen-associated molecular patterns, or PAMPs. They are conserved. The TLRs are conserved from plants to insects to humans. At the moment, there are 14 members of this family, and they are composed of leucine-rich repeats in the extracellular domain. They have a cytoplasmic domain, which is involved in signal transduction. You don't need to know that it's related to the IL-1 receptor family, but it's called the TIR domain. And the idea is that when the extracellular part of these molecules recognize a pathogen sequence, it then initiates signaling, which turns on the innate response. And these are some of the molecules that are recognized by toll-like receptors. They comprise bacterial and viral macromolecules, and in particular, TLR2, which is a plasma membrane TLR, recognizes some viral glycoproteins. You see a number of TLRs for recognizing RNA that is specific to viruses, double-stranded RNA, and then single-stranded RNA, TLR7 and 8. And TLR9 seems to be recognizing DNA specifically. This is what they look like. Again, they're transmembrane proteins. There's a cytoplasmic portion which is involved in signaling, a transmembrane sequence, and then the extracellular domain for each TLR comprises a different number of this leucine repeat. Now, the structure for TLR3 has been solved by X-ray crystallography, and the model is shown here. It's a curved molecule, and each of these segments is a leucine-rich repeat. So it's the same sequence repeated over and over. This is also glycosylated, and those are the little ball structures here. It has sugars attached to it. Now, the way these recognize PAMPs is either at the cell surface or within endosomes. So the TLRs that recognize viral glycoproteins typically recognize them at the cell surface. But there are many that recognize nucleic acid, and these are within the endocytic vesicle, because you'll remember that many viruses come into the cell via endocytosis. So when the nucleic acid is in the endosome, this is a great place to recognize it. So double-stranded RNA, single-stranded RNA, and also DNA are all sensed in this way by various TLRs. One thing that's a very interesting question here is, you may remember that when DNA viruses get into cells, most of them put their DNA in the nucleus. So we're not quite clear how those are sensed. And a lot of them don't go through endosomes. They just dock onto the nuclear pore and put their DNA there. So it's not clear how they would be sensed. But for RNA viruses, this pathway has worked out quite well. Once these TLRs sense the presence of the molecule, they then initiate a signaling cascade through a variety of adapter proteins, protein kinases, which ends up turning on gene expression, encoding for specific cytokines. And we have both inflammatory cytokines, which stimulate inflammation. We'll talk about that a bit. And then we also have the interferons, which will go, and both of these participate in trying to clear the virus infection. Here's just an experiment to show you what it looks like when you put an RNA into a cell and the cells respond. So this is an experiment where cells are treated with RNAs of various sorts, and the production of interferon alpha is measured. This is a cytokine induced by innate responses. And these are cells derived from two different kinds of mice. These are wild-type mice, and then these are mice lacking the gene for TLR7. You can take these genes away from mice readily, and they develop normally, but of course they're more susceptible to various infections. And you can see here that in each case you're putting different kinds of RNA into these cells. There's a very good production of interferon, but if you take away the TLR7 gene, which is the open bars, you don't have interferon alpha production. Now, if you put DNA into these cells, which is the last set of experiments on the right, CpG, a dinucleotide of DNA, you can see that even TLR7 null mice produce interferon in response. So it means something else is sensing DNA in these cells. So many, many experiments have been done like this to show what kinds of molecular signatures are recognized by these various molecules. So this is how these TLRs are believed to sense RNA. These are three different models for how the extracellular domain of the TLR is binding to RNA. The RNA is the coiled molecule here. So in some way, these bind RNA. And there are two interpretations of what happens. Some think that the TLRs be dimerized once they interact with RNA, and the dimer is most likely the active form. Others believe that these molecules exist already as dimers in the membrane and that the RNA binds to the dimer. In either case, once the RNA is bound, signaling initiates, and that leads to the production, again, of cytokines. Now, we also have, because not every virus goes through the endosome, we also have cytoplasmic sensors of nucleic acid, and two of them are shown here. PKR, you remember, is the protein that senses double-stranded RNA, and RIG-I is another one. There's a third one called MDA5. Yes?
The binding of the TLRs to the RNA produces cytokines?
Yeah, the binding of double-stranded RNA, or whatever ligand the TLR recognizes, leads to the production of cytokines by the cell, which includes interferon and tumor necrosis factor alpha, which induces inflammation. All right? We're going to talk about those specifically. These are cytoplasmic molecules, PKR, RIG-I, and MDA5, which sense RNA in the cytoplasm and initiate the same kind of signaling to produce cytokines by these cells. This is what happens with RIG-I, for example. This is a model of the activation of RIG-I by nucleic acids. RIG-I can be activated by either single-stranded RNA with 5' phosphates or double-stranded RNA. These are two RNAs that never appear in the cytoplasm of the cell unless it's infected by a virus. A 5' triphosphate RNA is never found. It's usually capped or has some other structure at the 5' end, and double-stranded RNA isn't found. So these are virus signatures. The RIG-I molecule is present in an inactive form in the cytoplasm. When the RNAs are present, they bind to the C-terminal domain. They unfold the protein, and then it can go on to stimulate signaling. And in fact, this N-terminal domain called CARD is an important domain for turning on the signal transduction pathway. So basically, the RNA activates RIG-I so it can initiate signaling. So here again, we have RIG-I, and the other cytoplasmic sensor is MDA5. And they sense various kinds of RNA molecules, and both of them initiate this signaling pathway. You don't need to know any of this here. The end result is the production of cytokines. This happens to be a very specific example, the production of interferon beta, but many other cytokines are induced when these proteins sense nucleic acids. The interesting aspect of this signal transduction pathway is that it requires a mitochondrial protein, which is called MAVS. And this MAVS protein binds the CARD domain of RIG-I or MDA when it's exposed after these are activated by RNA. And this has to be present in the mitochondria. If you make altered cells with MAVS floating in the cytoplasm, it doesn't work. And we don't understand why, but for some reason it needs to be in the outer mitochondrial membrane. Now, one of the most important set of cytokines produced by the innate system in response to these PAMPs are the interferons. And three of them are shown here. There are some others. There's interferon alpha and beta, which are called the type 1 interferons. These are produced by most cells in response to virus infection. And then we have interferon gamma, which is produced by T cells and NK cells. Interferons were first found in 1957 when two scientists found that if you infected chick cells with an influenza virus, they produced something in the culture medium which, when added to another cell, would protect them from infection. So they called it interferon because it interfered with the replication of influenza virus. And the first interferons to appear after a virus infection are the type 1s, alpha and beta. Interferon comes up later. The interferons are produced not only by cells that are infected with the virus. So when influenza comes into your respiratory tract, the initial cells infected produce interferon. But sentinel cells, the dendritic cells, macrophages that are in the area, will also produce interferon in response to what's released from the virus-infected cell. So if the cell dies, you have fragments of cells and viral molecules floating around. These sentinel cells will pick those up and sense them as foreign and begin to make a lot of interferon as well. So not only the infected cell, but the sentinel cells as well. This is something that happens very quickly after infection. Within hours, you get a burst of interferon, and then it goes back down because too much interferon is not good. And there are about 20-odd interferon alpha genes. They're all expressed in different ratios depending on the virus. There's just one interferon beta gene, which is on for a very short period. Now, interferon works on other cells to protect them against virus infection, and that requires that there are receptors on those cells for interferons. So here's a second cell which is being protected by interferons. Here, let's focus on interferon alpha beta. It binds a receptor. It's called the type 1 interferon receptor. And that binding of interferon stimulates a signaling pathway, the JAK-STAT pathway, which leads to the production in the nucleus of a variety of what we call interferon-stimulated genes. And this is the interferon response element, which is in the promoter region of these genes that are stimulated by interferon. So a specific subset of all the genes in us respond to interferon in this way. Again, interferon binds at the surface. You have a signaling cascade which leads to the transcriptional activation of specific genes. And those genes have the antiviral activity, not the interferons themselves. So we say is that the interferons induce an antiviral state. We get over 1,000 cell proteins induced, or mRNAs encoding various cell proteins induced. These are called ISGs. When they were first discovered, there were 10 of them, then there were 100, and only 2 years ago there were 300, and now there are over 1,000 induced by interferon. And these are the proteins that block virus infection. Most of them, in fact, we don't understand their mechanism of action. And probably different combinations work for different virus infections, so it's very difficult to study this and understand what's going on. So let's go through just a couple of these ISGs. Here's one that you should remember from the first part of the course, PKR, which is that protein kinase that's activated by double-stranded RNA. It phosphorylates EIF2 alpha, shuts off translation. Remember that this is a strategy for blocking virus replication, and it also triggers autophagy. So PKR is an ISG. It's an interferon-stimulated gene. There are also two proteins that act together to degrade viral RNAs. So here's an RNase L, which takes the viral RNA and chops it up. Good way to prevent viral replication. The RNase L, as it is induced by interferon, but it's not active as it's induced, has to be activated. It's activated by 2' 5' oligo A. It's a short stretch of A's linked in the 2 to 5' way. And those are produced by a second interferon-induced protein, oligo A synthetase. So these are both induced. This viral RNA activates the oligosynthetase. It makes oligo A, which activates RNA cell, and that chops up the viral RNA. So presumably this Baroque activation scheme is needed to ensure specificity. Another set of interferon-stimulated genes encode the MX proteins. These were first discovered in mice. MX1 blocks the ability of the influenza virus polymerase to steal caps from cellular mRNAs. You may remember that these caps are needed to prime synthesis of flu mRNAs, and this protein interferes with that step. But what is a bit puzzling about this gene is that there's no circulation of influenza in wild mice, yet about 75% of wild mice have the gene. It's not clear why, because they don't get flu unless we give it to them in the laboratory. However, there are two related human genes, MX genes. These inhibit, the gene products of these genes inhibit a couple of other viruses besides flu, VSV, measles, paraflu in the laboratory. What they do in us is not really known. We have no clue about whether these play a role in susceptibility or resistance to influenza virus, but it is an interferon-induced protein. Nitric oxide synthase is an enzyme induced by interferon gamma. This enzyme makes nitric oxide, and that has antiviral effects. Nitric oxide attacks proteins and destroys them, and it can kill virus-infected cells. And as we'll see in a couple of weeks, some of the pathology that we see with certain virus infections is a consequence of nitric oxide. PML proteins. These are proteins that are present in the nucleus. Did you mention PML proteins in your DNA? You did, and you're going to mention it again. These are promyelocytic proteins, or ND10 bodies, or pods. These are places where DNA replication of some viruses occurs. These can bind foreign DNAs and exert antiviral effects by altering transcription. And the proteins that make up these bodies in the nucleus are interferon-induced proteins. Now, these have antiviral properties, and as Dr. Silverstein will show you, many viruses encode antagonists of these PMLs to disrupt them because they don't want to be inhibited. The ubiquitin proteasome pathway has many components that are interferon-induced. For example, ISG15 is a protein that is conjugated to a protein when that protein needs to be degraded. It's a signal for degrading the protein. And that can be linked to virus proteins as a way of degrading them. The ligases, the enzymes that attach ubiquitin or ISG15 to virus proteins, are also induced by interferon. So the idea is that interferon induces this set of proteins with the idea of trying to degrade the viral proteins that are around. Here's a good example. If you inhibit proteasome, which is where these ubiquitin-mediated degradation events occur, you block the antiviral effect of interferon on hepatitis B virus, just as an example of what these do. A couple of other ISGs, ISG56, binds a subunit of EIF3, which is involved in protein synthesis, and it blocks translation. There are also microRNAs that are induced by interferon. These are small RNAs produced by the cell, most of which regulate cell gene expression, but they can also have antiviral properties. So, for example, interferon treatment of liver cells induces microRNAs which inhibit hepatitis C virus replication. This microRNA is very interesting, miR-122. This is actually a microRNA that is essential for replication of hepatitis C virus, which is a virus that infects the liver. About 3% of the world's population is infected with this virus. And interferon treatment actually reduces the level of miR-122, right? That makes sense because the virus needs it. Interferon reduces it to try and limit virus replication. Okay, so those are ISGs induced by interferon, part of the innate response. The other part, or another part of the innate response, comprises the sentinel cells, dendritic cells, and macrophages. These are cells that patrol the compartments of your body and look for trouble. Okay? Dendritic cells were first described as Langerhans cells by a man of the same name a long time ago. Ralph Steinman and Cohen called them dendritic cells for the first time in 1973. And you'll often see these referred to by one of two types, the myeloid and plasmacytoid dendritic cells. In your body, there are many immature dendritic cells patrolling the periphery in your skin, body cavities. These dendritic cells have TLRs on them. They have sensors in the cytoplasm, and they have receptors for cytokines. when they sense, say, products produced from a virus-killed cell, they become activated. They produce more cytokines, and eventually they go to lymph nodes, where they start to talk to the adaptive immune system. They start to have a conversation with lymphocytes of various types. And they say, there may be a problem here, we're not sure. And if there becomes a problem that the virus infection can't be cleared, then the dendritic cells tell the lymphocytes what's going on, and then eventually the lymphocytes will go out and check it out. So the dendritic cells make cytokines very early in infection. And they do that in order to try and limit the initial infection. So mucosal infection, the cells are infected, they produce cytokines. They attract dendritic cells. The dendritic cells get further activated by the dying cell debris. They make more cytokines. If they can't clear the infection, they will go to the lymph node and start to activate the adaptive response. So that's why we call them sentinel cells, because they're always looking for trouble. So on the left is a typical resting or unactivated dendritic cells. These have toll-like receptors on their surfaces. They have endosomes. They're highly endocytic. They take up material from the surrounding area, and they sample it to see if it's foreign. They can make interferon. They have cytokine receptors so that they're activated by cytokines. And they can take up proteins and present them in MHC class II molecules. Now, if they're near a virus-infected cell, the cytokines produced by those infected cells plus the debris will activate these dendritic cells, and they'll become mature. And now they look like a typical dendritic cell. They have these processes through which they can present antigens to lymphocytes in the lymph node. So here's a picture of one antigen being presented to a T cell, which will eventually activate it and have the ability to participate in the adaptive response. And again, these dendritic cells produce cytokines as well that activate and mature the T cells. Once activated, they not only can instruct T cells, but they can go on and elaborate a lot of cytokines to try and clear the virus infection. Now, as you might imagine, this is a very powerful component of the host defense. So viruses have evolved ways around this. So HIV binds a protein on the surface of DCs. So what do DCs do? What do dendritic cells do? They patrol. When they see trouble, they get activated and they go to the lymph node. So HIV hooks onto a DC. When it goes to the lymph node, that is where the CD4 cells are that HIV wants to infect. So it takes a ride on the dendritic cell to where it needs to go. Kind of nasty, but that's how it gets around finding the right cell types. Venezuelan encephalitis virus, equine encephalitis virus, replicates in DCs. So there are some viruses that actually can get in the DC and replicate in it, and that's bad news because it will destroy them and spread the infection further. A number of labs are looking at this as a way of getting virus to the nodes, because that stimulates a very good antibody reaction. And if your virus doesn't kill the cells, maybe that would be a vaccine strategy. And as I said, if you infect DCs and destroy them, you can interfere with the maturation of the DC, migration, activation of T cells. So there are examples of interfering with DC function at every step that are imposed by virus infection of these sentinels. So they're not always infected by the virus, but when they do get infected, it's trouble. Another component of the innate response is complement. This is a very old system, at least as far as we know it, identified in 1890 as a factor in serum that would lyse lyse bacteria as long as antibody to the bacteria were present. So they said it complements the activity of antibodies, so it was called complement. It's a major defense system that sort of bridges innate and adaptive systems. You'll see it composes a lot of different proteins which help to amplify the innate response. It's basically a collection of proteins that are in the serum and in cell membranes that achieve this purpose. And it can recognize invaders similar to the TLRs and the cytoplasmic sensors by pattern recognition. It can tell a pattern in an invader and then use the antibodies that are being produced by the adaptive system to further attack the invader. There's also an example of some natural antibodies that we have against a variety of bacteria, which we always have in us, and this complement system takes advantage of those before the adaptive system can even kick in. There are 4 functions of complement. It makes holes in infected cells, cytolysis. It activates inflammation. It can stimulate the production of cytokines, which lead to inflammation. It can help coat particles, virus particles, bacteria, to facilitate them being taken up by a cell to destroy them. And it can solubilize an immune complex. So when you have an antibody response to a pathogen, you have antibody-pathogen complexes form. And if you have too many of those, which can happen, complement's job is to break them up so they won't be deposited in very small capillaries like those in your brain or kidney, and that causes big problems. This is just an example of the pathway to show you how it's sequential and amplified. We start here with one of the components of complement, the C1 component, which will recognize an antibody bound to a viral protein on the cell surface. So let's say this is a cell infected with influenza virus. The virus is putting HA and NA glycoproteins on the surface, getting ready to make new virus particles. If there are antibodies to these glycoproteins present as a consequence of the adaptive response, complement will recognize them, and it stimulates a whole cascade of reactions, which leads to the formation of this membrane attack complex. And this will poke a hole in the cell and kill it. Okay, so just one of the activities of complement which helps both the innate and adaptive system get rid of pathogens. So the innate system comprises cytokines, pattern detectors like the TLR and the RIG-I, NK cells, complement, the sentinels, macrophages, dendritic cells. And these take care of most infections before we know it. Most of your assaults with viruses on a daily basis are taken care of, and you have no sense that anything is going on because you don't get fever, you don't get any of the signs of inflammation. But if the infection goes beyond that, which, as you know, happens, the innate system says, I can't handle this, and goes to the adaptive system and kicks it in. And that's one of the main jobs of the dendritic cells and the cytokines that are produced by the adaptive system, the innate system. So the cytokines that are produced, we've been talking about cytokines induced by the innate system for some time. There are 3 main classes. There are pro-inflammatory cytokines, which we'll talk about in a moment. They produce all the classic signs of inflammation. There are anti-inflammatory cytokines, because you can't keep producing inflammatory cytokines. You would have an overbalanced reaction, so there has to be a balanced system. And then there are chemokines, and these induce inflammation. They recruit cells to the infected area as a way of trying to clear it. Now, when you get a virus infection, you get a fever, you get headache, you feel hot, you get muscle pains. None of that has to do with virus killing your cells. All of that is a consequence of these pro-inflammatory cytokines. So, for example, IL-1, TNF-alpha, IL-6, one of the targets of these is your brain. It makes you tired. It makes you have a fever. It makes you ache. These are systemic symptoms, and they can be caused by a localized respiratory tract infection. And that is, again, a typical sign of a cytokine. They can diffuse everywhere in the body and cause these symptoms. Cytokines also go in the liver and induce a variety of other proteins, which we don't have time to go into, which also have antimicrobial properties. They also go into the bone marrow and stimulate the production of lymphocytes, which is what you need to make antibodies and to destroy infected cells. So the cytokines produced have a global effect on you. And just remember, the next time you have a virus infection, you can ascribe those symptoms to these cytokines. But they're doing the right thing. They're making an inflammatory response. So these cytokines induce what we call an inflammatory response as the intrinsic defenses are activated. TNF-alpha is a major part of this, but there are other cytokines that participate as well. And there are 4 classic signs of inflammation: redness, heat, swelling, and pain. And again, these can happen globally as a result of cytokines diffusing from a primary site of infection. These symptoms are a consequence of many different things that go on: increased blood flow caused by the cytokines and products of cells that come into the infected area, increased capillary permeability so that cells can come in and out easier, phagocytic cells come into the infected region. There's tissue damage. So many things contribute. But the cytokines are the major initiators of this inflammatory response. So the main take-home message is that this has a global effect. The innate response to a respiratory infection in your upper tract has global consequences because of the cytokines. And it involves a cooperation between the lymph system and the circulatory system. So if you have a pathogen that has come in and infected your respiratory mucosa, it produces antigens that are recognized by cytokines. And as I've told you, that are recognized by dendritic cells. These produce cytokines that recruit other cells into the area, and those cells produce additional cytokines which amplify the response. You have migration of cells such as macrophages from the blood into the area. The permeability of the bloodstream is affected. And if the infection can't be stopped at an early stage, the dendritic cells go into the lymph circulation. They go to the nearest lymph node and activate the adaptive system, that is the T cells and the B cells. So this is a very interesting correlation now. Inflammation is good for you, even though it makes you feel bad. It's good because you get very good immune responses. When you have a really good inflammatory response, you're likely to make good antibodies and T cells against whatever is infecting you. Now, cytopathic viruses, the ones that kill your cells, that lyse them, they are really good at causing inflammation because they break cells open and the dendritic cells take up the bits and they get stimulated and activated and make a lot of cytokines. You get great inflammation. And inflammation, the production of cytokines, is key for activating the adaptive response. Now, so these guys are really good at activating the innate response. And because the inflammatory response is so effective, many viruses have proteins that modulate it. And Dr. Silverstein will tell you about some of those next week. Some of the big DNA viruses have interesting proteins that counteract these cytokines so you don't make an inflammatory response. Some viruses don't activate the adaptive response well because they don't stimulate good inflammation. You get infected with them, you don't get fever, you don't get aches, you don't get pain because they are not very good at inducing an inflammatory response. They're typically non-cytopathic viruses, that is, ones that don't break cells open. Cells aren't damaged or killed. There's no cytopathic effect. And you have a very poor innate response, low cytokine production, and low inflammation. So these are really different in terms of their interaction with the immune system. And again, Dr. Silverstein will talk about those when he goes into persistence. So these viruses that don't induce good inflammation, like measles virus is one of them, they may cause persistent infections and not be readily cleared. So the lesson is this classic inflammatory response is great, and it reflects the fact that the innate and adaptive systems are communicating. Viruses that are good at inducing an inflammatory response make a really nice adaptive response. If you don't get a good inflammatory response, you get poor antibodies in cells. And this is the reason why we want to put adjuvants in vaccines. So many vaccines, the flu vaccine is a great example. You take virus and you inactivate it with formalin and detergent. You break it up. What you get injected with is a little bit of protein. There are no particles. There's hardly any RNA in it. It's a really poor inducer of inflammation. You get an immune response, but it could be a lot better. And in Europe, they mix it with adjuvant because adjuvants, which are typically oils or other materials added to the vaccine, stimulate inflammation. They tickle the innate response so that you get these cytokines made, and that leads to a good adaptive response. But we don't use them here because people think they're dangerous. Okay, so let's go back. Let's go into the adaptive response now. And what we've discussed so far, for example, we have infection of an epithelial barrier with a virus. These infected cells make cytokines. The cytokines attract dendritic cells, and they mature. The dendritic cells make more cytokines, and then if the infection isn't controlled, the dendritic cells go to the lymph node and start talking to the adaptive response. And then you make antibodies and cells to try and clear the infection. And you, many of you have had Dr. Moshowitz's immunology course and learned all of this. For those of you who haven't, there's a pretty good summary in the textbook. We're going to talk about two arms of the immune response. The arm that produces antibodies, that is, viruses interact with B cells and stimulate them to produce antibodies that will neutralize the virus. And then the other arm, the cell-mediated arm, where you make T cells that are specific for your pathogen. And some T cells are specific for making cytokines that assist in the maturation of B cells or the maturation of the other kind of T cells, the ones that actually lyse infected cells. Adaptive responses, which we just described, are essential for getting rid of an infection, for clearing it. An innate response can stop it early, but once you've got an established infection, you need an adaptive response to clear it. And the effectors are antibodies, as we've said, made by B cells. These bind to viruses, either in the blood or at mucosal surfaces, decrease the number of viruses, and limit the infection. T cells, which also recognize infected cells, and they see antigens on the surface of the infected cells. And there are cytotoxic T cells, which kill virus-infected cells, and then helper T cells, which we've said produce cytokines that assist in the maturation of these lymphocytes. Now, the innate immune system, you remember, distinguishes foreign by means of these pattern recognition receptors. The TLRs, the Rig-like helicases, MDA5, complement even has pattern recognition components that we didn't talk about. NK cells do a similar thing. They recognize altered self receptors. But the adaptive system does it differently. And we don't have time to talk about this, but they rely on antigen presentation of an infected cell to know that it's foreign. So they distinguish an uninfected from an infected cell by the presence of viral proteins on the surface, which are presented by the major histocompatibility complex proteins. So T cells and B cells both do this. Antibodies are an essential product of the adaptive response. Many virus infections induce antibodies, and these do what we call neutralizing virus particles in the blood. We'll talk about that in a moment. You can have antibodies made at mucosal surfaces. They can be produced in the blood, and they're secreted across the epithelium and end up on the mucosa. So when a virus lands there, it's blocked by the antibody. Or you can have antiviral antibodies in the blood. And in general, we think that antibodies are important for preventing infection, and cell-mediated responses are important for recovery. But there are exceptions, like everything else in science. So some antibodies actually help in recovery and vice versa. So here's an example of how an antibody can protect you against infection. This is an experiment where animals were given antibody to poliovirus, different dilutions, and then they were injected with poliovirus, and the percent paralyzed animals was determined. So here, if you don't do anything to the animals, the control, you just inject them with polio, they all get paralyzed. If you give them antibodies, if you give them very dilute antibodies, it doesn't help them, but increasingly concentrated, you see, protects them against paralysis. So these antibodies are getting into their bloodstream and blocking the spread of virus infection. How do they do that? There are many ways that viruses block infection. You remember from early on in this course, we talked about viruses binding receptors and being taken up into cells, perhaps by endocytosis. So there are some antibodies that will actually block attachment of the virus to its receptor. But there are also antibodies that block subsequent steps. So there are some antibodies that get taken up with the virus into the endosome, and they prevent the uncoating of the virus in the endosome. And then there are also some antibodies that cause particles to aggregate. And so in that state, they're not infectious. So 3 different ways of neutralizing virus infectivity. There are very specific parts of viruses that combine with antibodies. And since we now have structural information at high resolution for many viruses, we can see where these are located. So on the left is a rhinovirus particle, and all the light-colored areas are sequences or epitopes on the particle where antibodies bind that block infection. So the sequence that an antibody binds is relatively short, and we can identify these on the viruses in a variety of ways. And you can see these are in very defined places on the virus particle. This is the hemagglutinin of influenza virus. You recall that this virus, this glycoprotein attaches to sialic acid, which is the cell receptor. Here's a top view of the trimer of the hemagglutinin. The sialic acid binding site is in here, and surrounding it are areas where antibodies that block infection bind. So these antibodies block influenza infection by preventing it from attaching to its receptor, sialic acid. And it makes sense because their binding site, which is, maybe you can't see well, but it's light blue compared to the rest of the molecule, is surrounding the sialic acid binding site. Okay, so cell-mediated immunity, on the other hand, is what is probably the most crucial for getting rid of an infection once you have it. And there are different components of the cell-mediated response, which we'll talk about. There's type 1, the Th1 response. There's cytotoxic T cells, which lyse infected cells. And we have lysis of target cells, that is virus-infected cells, by transferring granules from the T cell to the target cell, which basically cause the lysis of that cell. And these granules contain a protein which pokes holes in the target cell, and also enzymes that help digest the target cell away. And many CTLs also induce apoptosis in target cells when they recognize them, and that's another way of killing them. So here's a movie of a CTL, which is this guy here, and 2 target cells. And he's looking at this one but not doing anything, and then this one he hits and he kills. So this CTL was full of, all the granules in this CTL were full of a red dye, and then that red dye is transferred to the target cell. And in the process, the green dye, which was preloaded in the target cell, leaks out. So this T cell, this CTL, recognized this one specifically, poked holes in it, transferred the dye, and then that cell became permeable. Now, one of the interesting aspects of this is that the CTL doesn't do anything to this cell because it's seen as self. It doesn't have viral antigens on the surface. But this one does, and as soon as it recognizes that, it kills it. So it's very specific. So this cell is not infected. It doesn't display a viral antigen. This one does, and it's recognized by the CTL. In some infections, both antibodies and cells, T cells, play a role in host defense. And here's one example of that. This is infection of animals with lymphocytic choriomeningitis virus. And we're looking at the level of virus in serum, spleen, and brain. So normally, let's look at the control animals. You inoculate the animals with virus, about 30% of them die. This is the time course of virus replication. It goes up, it peaks at about 5 days, and then it goes down. If you take away cytotoxic T cells, which you can do experimentally, that's the green line, doesn't change the course of virus growth in the serum. But if you take away antibodies to the virus from this animal serum, you can see you prolong the persistence of virus in the serum. So the animal can't clear the infection as well when you take antibodies away. If you look in the spleen, here's the wild type or the control pattern of virus production, the dark line with the circles. If you take away antibodies or cells, you prolong virus replication. So in the spleen, both are important for clearing infection. As you see also in the brain, here is the untreated animal. You take away cells, CTLs, or antibodies, the infection goes up. So in this case, both antibodies and cells play a role in defense against infection. And here's an example where antibodies are important. So these are monkeys infected with monkeypox, which is basically the smallpox of monkeys. These are all the animals are first immunized with smallpox vaccine, the same vaccine we would get. It cross-reacts, cross-protects against monkeypox. So on day 0, you immunize the monkeys, and then you do some manipulation. Here's the control group. You don't do anything to them. 22 days later, you see very high levels of antibody against the vaccine virus in their blood. You then infect them on day 28, and then none of them die. So they're protected by this antibody in the blood. If you immunized another group of animals and deplete their B cells at 4 times in the course of the experiment, They make much lower antibody in the blood, which you would expect because the B cells are making the antibody. And then when you challenge them with smallpox, 3 out of 4 of them die. So the antibodies are essential for protecting them against infection. If you take away their CTLs, their cytotoxic T lymphocytes, this has no consequence on whether they survive or not. So this is an example of the antibody being essential for protection against infection. Now, there are other examples where CTLs have been shown to be important for resolving an infection. So an animal gets infected, and the curing or the resolving of the infection depends on cytotoxic T lymphocytes. So this is an animal model of hepatitis B virus. You infect the animals with hepatitis B, and then you sample the animals, and this is a gel where we're looking for replication of the viral genome. So this band means that the virus is replicating in the animal. So if you treat the animals with just sodium chloride, the virus grows because this is a control. If you give cytotoxic T cells to these animals from another animal that had recovered from a Hep B infection, so now these are Hep B specific cytotoxic T lymphocytes, you protect the animals from being infected. You see no viral genomes here. So that tells you that CTLs are important. But the second experiment is very interesting. They redo the experiment. They infect the animals with hepatitis B, and then they give them the same CTLs, except they're now mixed with an antibody against interferon gamma. And now you see these animals all become infected. So that tells you that the CTLs protect the animals, not because they lyse infected cells, which I showed you a movie of just before, but they secrete interferon gamma, which apparently is important in protecting them from infection. So again, the moral is that some viral infection, the clearance by these cells depends on cytokines, not the cytolytic activity of the CTLs. For some infections, as you might gather, the CTL response is more important, or vice versa. That's what I've been trying to tell you. How do you determine the combination that's produced? Well, it all happens here in the lymph node. When the dendritic cells interact with the T and B cells, the particular mixture of cytokines that are produced by the dendritic cell instructs the T cells which way to go. More antibody or more cellular responses, and it depends on the virus. So the dendritic cells produce a cocktail of cytokines depending on the infection. This production of cytokines is influenced by the kinds of T cells that are produced. And many of you know that there are two kinds or two subsets of T cells, T helper cells, Th1 and Th2 cells. Remember, the T helper cells, not the cytotoxic T cells. The T helper cells make contact with the dendritic cells in the lymph node, and they make cytokines that help mature B and CTLs, B cells and CTLs. So the information that these helper cells get from the sentinel cells determines whether you get mostly antibodies or mostly CTLs or a mixture of the two. And that is because the Th1 and Th2 cells make different kinds of cytokines which can influence that production. So Th1s make cytokines to help mature CTLs, and Th2s make cytokines that help mature B cells, the cells that make antibodies. So again, whether you go predominate Th1 or Th2 depends on the infecting pathogen and how it's programmed the dendritic cell. So Th1 cells turn on killer cells, and most virus infections get cleared by this response. Th2 activate the antibody response. This is usually not a good response for most viral infections, with a few exceptions, because most antibody responses can't clear a virus infection. So which response gets turned on? And as I said, it's the mixture of cytokines that are produced by the infected cell, by the dendritic cell. As it goes into the lymph node, it instructs the system. So you have an antigen-presenting cell in a lymph node, a dendritic cell that has sensed an infection at a mucosal surface. It goes into the lymph node, and there are immature CD4 cells there. And it's going to tip the balance either to Th1 or Th2, depending on the virus and the kinds of cytokines it's encountered. And Th1 will produce a certain cocktail of cytokines, and Th2 will produce another that give either CTL or B cell preference. And we don't really know how the decision is made. One hypothesis is that the Th1 predominates unless the dendritic cell instructs otherwise. So in other words, make a CTL type cellular response. And if there's a particular virus that needs antibodies, then the dendritic cells will instruct, so forth. The other hypothesis is that the TLR detection of RNA leads to a Th1 response, where you get mainly CTLs. But we don't really know at this point which is right. Now remember, adaptive responses give you memory. So there's a lot that goes on, but if you forget it, it's of no use, because you're going to be infected again if you survive, so you have to remember what to do. So if you get infected, the response is rapid and it's specific. It takes a lot less time to make antibodies or CTLs once you've had the initial encounter. Innate responses don't have memory. They turn on very quickly and respond to the particular pathogen. They don't need to have memory. They're tailored to be rapid already. And of course, memory is why vaccines work. You get immunized and your immune system remembers that antigen or collection of antigens, and when you see it again in terms of a virus, you can respond quickly. So here's an example of memory. This is the time course of production of antibodies in the serum in response to two different antigen inoculations. So let's say we inoculate antigen A here. You get a primary antibody response to antigen A. It takes a few weeks to develop, typically 3 to 4 weeks. It's not so robust. The titers are low, and then it goes away. The serum antibodies go away. But if you get reinfected, then you have a very robust secondary response. In this experiment, they've actually injected the animal with a mixture of antigen A plus B, so you can distinguish the secondary anti-A response, the memory response, from the primary anti-B response. So this is much quicker, you see, and it's of higher level. You have higher titers of antibodies in the serum. So memory is characterized by a specificity, higher titers, and faster response. So that was the memory of antibodies, but there is also cellular memory. So when you are infected and you develop a cytotoxic T cell response against the virus infection, there remain also within you memory CD8 cells that can also respond to a subsequent infection. And that's shown in this experiment. Here, animals have been infected with a virus, and at different days after infection, measuring the virus in the spleen. This is lymphocytic choriomeningitis virus. You have a peak of virus production, and then it declines. The bottom panel is measuring the CTL response. So these are virus-specific CTLs that can lyse infected cells. You can see these peak correspond just after the virus peak, the virus titer peaks. You have a peak in the CTLs, that is cytotoxic T lymphocytes that can lyse virus-infected cells. These go away relatively quickly with the same kinetics as the disappearance of virus. But if you look for CD8 cells that can't kill infected cells, but simply can recognize proteins from the virus, and there's a separate experimental way to do this, you can see that those persist for a long period of time. So these are memory CD8 cells that are specific for this virus. If you are infected again with the same virus, you will have a very rapid elaboration of CTLs that can kill virus-infected cells.